Today I chat with Melinda Kliegman, who is the Public Impact Director of the main CRISPR Center in the world at UC Berkeley. It's a great conversation. And it's part of the series that we're doing on rewriting all of biology and all organisms. And in today's episode, one of the really cool things we talk about is how important public education is for CRISPR. And, Melinda talks about this crazy thing where our status quo right now, we grow up into a status quo with nation states and computers and those things being true. But obviously that wasn't true 500 years ago. And now even the Gen Z kids grow up with TikTok. They are what we call digital natives. And in the future, we're going to need to become kind of CRISPR native, where we grow up into a society where CRISPR is true. And that's going to be a big shift around public education that we need to do for this new technology. So enjoy the episode. Hello, listeners. My name is Rhys. I'm the founder of Root, and welcome to The Rhys Show. Today, I'm excited to chat with Melinda Kliegman. Melinda led bioinnovation policy and advocacy at the Gates Foundation and is now the Public Impact Director at the Innovative Genomics Institute, which is this cool new institute from Jennifer Doudna, which is all about kind of applying and looking at research, CRISPR research. And it's at UC Berkeley and at UCSF. Melinda, thanks for being on the show and welcome.
Thanks so much for inviting me, and I'm excited to talk with you today.
Yeah, I think it'll be fun. And Melinda and I were chatting before about how she was a a biotech kind of bio PhD back in the day, but it's also been 10 years for her since being on the bench doing that, and now she focuses a lot on public policy. And so that's what we're going to focus on today. Is a lot of how this kind of new biotechnology, especially CRISPR technologies, are going to be good for society, hopefully. But before we get into that, Melinda, I want to ask you, for you, what is this through line that ties your work together? You got this PhD in biology, but you're interested in this public impact role. How did you kind of navigate through that transition?
Yeah, I think, two of my core values that underlie a lot of my professional decisions. One of them is definitely a love for nature and just general curiosity. And then I think there is also a really strong driver to have a career in public service. So I was born in Jamaica and I spent most of my formative years in Guyana in South America. And it's a fairly impoverished country, but it's also home to a portion of the Amazon rainforest. So I grew up, spending a lot of time outside, climbing trees, avoiding giant centipedes, eating random fruit and hoping it wouldn't make my mouth super itchy. And I was there because my dad was a minister. So I also spent a lot of time in service to my community. And so I think at the core, those are sort of my two most elemental values. And with my PhD in ecology, I wanted to do that because I really wanted to learn more about the natural world and be outdoors. But I had a difficult time with it, I think, because when you're doing basic research, you're really pushing on the boundaries of knowledge and trying to understand for the sake of understanding. And I think that is what makes science a really noble profession. And not everything needs to be applicable to advancing humanity. But for me personally, that kind of research didn't really jive with those core values around public service. So after my PhD, I went to the USDA and then the Gates Foundation and now the IGI because I want to use what I know about biology in service of people.
Cool. Yeah, that's a great, concise and clear answer. That's a cool, it also makes me think about, for me, I remember I was a computer science major in college, and I remember taking I wasn't sure whether I wanted to go into research or whatever, and I took a class on advanced algorithms or whatever, and it was supposed to be a graduate level thing. And it was just, it was doing these tiny little things that were oh, can we make this algorithm 0.2 times, more efficient? And I was kind of bored by all of it. And again, as you say, there's nothing wrong with that. Some people are into pushing the bounds of knowledge, but maybe you and I are more about okay, how does this going to impact people?
Yeah. I think it's very important for me.
Totally. So talking about your role at IGI, let's kind of dive into that for a bit. And I think that, just to give our listeners some background here, it's a crazy thing. CRISPR was only discovered, less than 10 years ago. And now there's going to be all these implementations of this new CRISPR technology in the world. And IGI is one of those places where that's happening. So could you tell us a little bit more about, you guys have human health work, you have climate work, you have just advancing human, genomic engineering work. Could you tell us a little bit more about what IGI does in each of those areas?
Yeah, sure. I mean, there's so much going on. I think this year is actually the 10th anniversary of the discovery of CRISPR, so that's pretty exciting. I mean, in human health, one of the things that's happening is a clinical trial for sickle cell disease. So that was approved by the FDA last year, and it will likely be starting this year and running for 4 years. And researchers using technology developed at the IGI. It's being run in conjunction with our partners at UCSF and UCLA. And they're using CRISPR-Cas9 to correct the mutated beta globin gene that causes sickle cell disease. And then in conjunction with that, my team is looking at ways that we could make any of these genetic therapies more affordable and accessible. Our trial is one of, I think it might be the only trial that's being run by a university nonprofit. And what we're seeing is a lot of these genetic therapies that are coming on the market, they are coming on for price tags that range from 400,000 to 2.2 million per patient. So we're trying to see, what can we do as a public academic institution that gets a lot of our funding from, the NIH or the California Institute of Regenerative Medicine. So sort of taxpayer dollars that funds research. How can we deliver our innovations to society at a more reasonable price tag? So my team has spent the last few months assembling experts from insurance companies, government agencies, contract manufacturers, social venture capitalists, public benefit corporations. We pull them together in what we're calling an affordability task force
Mhm.
to see what this motivated group of experts can come up with as a creative solution to this problem around affordability, so we can take things that are being developed in our research pipeline and plug them into this hopefully new creative solution. So that's sort of the human health space.
Wait, actually, can I pause
Yeah,
you for a second before you
of
go? Because
course.
that's all delightful, juicy stuff. So yeah, you have this sickle cell thing, aka yeah. And if I understand it correctly, it's like we have our little cells in our bodies, but they're usually circles, and the sickle cell means it's a moon shape or whatever. And actually, for malaria, it's good to have this kind of sickle cell thing because then you can kind of your body's like good at attacking or like fixing its blood cells or whatever and from a malaria perspective, but it's bad generally because the blood can't hold as much oxygen or whatever, and so you're going to go in and CRISPR is going to change the kind of the gene that creates the blood cells or something. Is what's tell me more about that.
Yeah, so it's a monogenic disease. So it
Okay,
means it's caused by mutations in one gene.
cool.
And what's fascinating is that it's actually that same mutation has arisen multiple times in our evolution as a reaction to malaria pressure. And so what you're saying is exactly correct. If you have one copy of that gene, then you are somewhat protected from malaria. But if you have both copies, it's from both parents, then it's really deleterious. You can have, strokes at a young age, severe pain. So and you have a shorter lifespan as well. So what researchers at the IGI are going to try to do in this clinical trial is you basically take some of the cells out of the person's body. You can use CRISPR to directly edit those cells to correct that mutation, and then you grow them, so you make some more of them, and then you put them back into the person. And if enough of those take, then that person will start to experience fewer of those very negative symptoms associated with sickle cell disease, because they'll be able to make some healthy blood cells.
Cool, got it. And so the cells, are you taking out red blood cells and then putting them back in, and then but I guess the body itself is still creating the sickle cell ones, or do you have to like do that process often to like pump back in the new CRISPR cells, have red blood cells?
No, so it should hopefully be only one time.
Okay.
So you take out hematopoietic stem cells, and you make changes to those. So stem cells are the cells that can continue to replicate and make more of themselves. And then those are the ones that you edit and put back in. And you don't need it to be all of the cells. I can't remember the exact percentage, but you just need some percentage of those cells to be corrected, to be able to experience the positive effects, basically.
Cool. Got it. That's interesting. Okay, got it. Yeah, so that's on the sickle cell side. And then as you were saying before, the other interesting thing of what you all are doing is that yeah, there's obviously tons of, CRISPR stuff happening in the world. And what you are doing is you have this kind of public, you're a nonprofit academic kind of crew. And so how do you that affordability task force, how do you make stuff more affordable? Or is it, part of the thing is just like a market thing where it's like maybe they're charging $2 million because it's CRISPR's expensive or whatever. I don't know.
Hmm. Yeah, I mean, I think that is one of the things that we are trying to figure out. What exactly does it actually cost? Like, there isn't a lot of information out there about it. So we're not sure, like, is it truly cost this much to make it, or is it just a significant markup? And I think it's probably a bit of both. Like, it is probably expensive to manufacture and deliver the therapies, but it's also companies that are trying to maximize profit for their shareholders. What we've also seen too is, the IGI also works on rare diseases. Some of the companies have licensed technology developed by researchers and academic institutions, and then realize, oh, we can't make enough money from this because not enough people have this disease. And then they've just like shelved that therapy and no longer developed it when people out there could benefit from it and use it. So, in some senses, the capitalistic model be may not work for some of these therapies.
Yeah.
And so that is something that we don't have the answer, and we're embarking on this process to try to figure out what, if anything, can we do to make the situation a little bit better than it is today.
Yeah, that makes sense. It is funny because you say that, and I'm reminded of, I was looking up the drugs in the world that they're the ones the most profitable, or either profitable or the ones that have the highest revenue per year, and they're all diseases that affect lots of people but also don't go away. So like rheumatoid arthritis is like the main one where it's like you're going to have it for a long time, and you're going to be able to get a lot of money out of that drug because it's going to versus something where it's like if you prevent the person from going into the hospital in the first place, you don't get any money for that. And so like yeah, those are kind of larger issues perhaps within our system. One other
Yeah.
question, or yeah, what were you gonna say?
Oh, I was going to say, I think that companies build some of that into the price tag because some of these genetic therapies are potentially curative, and someone will only need one treatment. Then it's more highly valued. And so they can justify the higher price tag by saying, oh, this person's going to derive a lot of value from this. And so will the healthcare system as a whole, right? Because you don't have to pay for that person to keep coming back. So $2 million is a great price tag based on that. But, that that doesn't really jive with our sense of public service. So,
Totally.
yeah.
Yeah. Do you think that there's this is just like a thing that I don't know the answer to, which is I know when you see curves for like Moore's law style curves for sequencing and synthesis, it's like, oh, it used to cost obviously in Sanger sequencing way back in the day, it was like so expensive to sequence genome, and now it's like
Mhm.
I forget the amount, but it's essentially free. I'm not free. It's $300 for my genome, which is very
Yeah.
cheap. And then synthesis is also now it's like 8 cents a base pair or something. And so it's how much does CRISPR cost like per base pair or whatever?
Um, okay. Okay, that's a little an interesting question. So I think it's just really dependent on what you're trying to do, right?
Uh-huh.
And even with sequencing.
Hmm.
Um, so how complex is the system that you're looking at? And I think there's a wide variation in cost. So let's take, you mentioned sequencing. Let's take that for an example. If you're looking at the entire genome of a complex organism, so like wheat, the wheat genome has 15 billion base pairs. And it took like a team of researchers 15 years and like $75 million to sequence the whole thing. Versus, a virus or a bacteria where you could get the results in a day for a few $100s. And I think it's the same thing with CRISPR. So if you are trying to run like a high school lab experiment, what's a proof of concept using bacteria, you just want to make them glow using CRISPR, that might be several $100s. or it could range into the millions if you're trying to learn new information or work in a more complex biological system. So I think there's a really wide variation, even in between any of those technologies.
Cool. That's helpful. But even it's cool to hear that, my girlfriend is a biology teacher, and she could, if she wanted to, try to use CRISPR in her classroom for a couple hundred. I mean, it'd be expensive, maybe only one one thing per year, not every student. But that's cool that you could do that. So let's kind of
Yeah.
transition to the other side here. So yeah, you're talking about public health. What are you guys doing in climate?
Yeah. Oh, wait, I just want to say one more thing. I
Yeah.
think the IGI has developed a CRISPR kit in conjunction with the Lawrence Hall of Science at UC Berkeley. And I don't think it's for sale quite yet, but it will be really soon. And it's for high school teachers. They get a kit that they can use with their entire class to learn
That's
how to do
amazing.
CRISPR.
That actually makes me one other point on that, which I think you all did a really good job of, is just shaping the public educate. Like you made this recent book or something like that of like teaching CRISPR for kids, and it was like a really
Mhm.
cool. Let's actually talk about that for a second while we're still going on it. What like I thought that book was, and I think part of your job is this public education. Part of that's like public impact, and we're going through this pretty intense transition where it's like we're going to be changing our genomes and stuff. How do you think about that in terms of shaping a narrative or an educational system around CRISPR and its impacts?
Yeah, I think having penetration into society where people know what something is, they're interested in it and want to learn more. I think that is such an important component of this. Like you said, we are potentially making changes to our biosphere, to ourselves. And I personally think that it's really important for as many people in society as possible to be engaged in that dialogue and that discussion, um, particularly people who have been traditionally marginalized or are not, generally asked what their opinions are. We're also translating a lot of our educational materials into other languages so that it can be more accessible. So a big part of our institute as whole is this public impact thing. I think this is what makes us special in that we are thinking about ethics, we are thinking about public policy, we are trying to engage the public, we are providing educational materials as much as possible.
Yeah, I think that's amazing. I think it is such a crucial. It's like how can there's this big new change happening, and how can we kind of incept the I mean, incept the population is not the term that one wants to use, but like how do we make it? How do we create this thing where everybody kind of knows? Okay, this is what's actually going on here, and now let's have a talk about how we should use it and all those kinds of things. So, okay, beautiful. So let's
Okay.
talk about climate. What do y'all do with climate stuff?
I mean, we have a fairly large climate program, and it kind of looks across the carbon cycle to see what changes we can make or edits we can make to sequester more carbon. And there's also some projects in climate adaptation as well. So I'll give you an example. One of our IGI researchers, Pam Ronald, has created a large library of rice mutants that have longer and deeper roots. And so the world's agricultural soils have lost something like 487 gigatons of CO2. And so deeper roots are an excellent way to put carbon further down in the ground where it's more persistent and more recalcitrant and doesn't just get like plowed back or returned into the air. And those longer, deeper roots also have added benefits like making plants more drought tolerant, for instance. And then we're also looking at things like how microbial communities interact with the roots to improve carbon storage, and whether there's like changes and edits that you can make along the way to enhance any of those naturally occurring processes.
Cool. And so those roots, and again, it's kind of funny because it's just like the same little tool. Is that also just like you take out the thing, you say, okay, this is what the plant, here's a little gene for plant root, and that one's more polygenic probably, I don't really know. And then you say, hey, let's edit some stuff, and boom, you got longer roots. Or is that one different than the sickle cell one, or no?
I mean, I think the great thing with plants is you can kill them.
Yeah.
It's okay. So I think, researchers sort of you can knock out or knock in a whole bunch of different genes and then observe what phenotype that creates. So are they longer? Do they have more branches? And then you can pick ones that have specific traits that you're really interested in, and then continue to work on those specific genes or breed them together. And so you can kind of do a screen, is basically what you're trying
Yeah,
to do.
but poor little plants, so they're getting hurt, you know? No, yeah, I mean, the plants will be fine. The plants will be fine. Yeah.
Yeah, that's why I went into ecology as opposed to like anything to do with the human molecular biology. I'm like, oh yeah, the mistakes are a little bit easier to swallow.
Yeah, that's good. Yeah. It makes me think too of, yeah, you have, so yeah, and I guess one other question there. See, are you changing? Yeah, okay, no, I think that makes sense to me. And I guess maybe then the final kind of big pillar that you all have is this, beyond the public education impact one is, advancing genomic engineering itself, because CRISPR was the first, like, here's random CRISPR, but then like we're gonna get better and better and better at CRISPRizing things or whatever. So tell me a little bit
Mhm.
about that.
Yeah, so I think Jill Banfield and the work that's happening in her lab is just really cutting edge and very cool. She continues to just add to the tree of life of new organisms that we didn't know existed before. But some of the researchers in her lab are using CRISPR to directly edit microbes in a mixed community. So first, researchers are able to identify the bacteria in the community using a sequencing approach called environmental transformation sequencing, or ET-seq for short. And then they can go in and directly edit specific microbes in, let's say, a 200 member community. This is still happening within the lab. And so this is really cool because in the past, you'd have to culture microorganisms. So I did that a lot in my own research. You take an environmental sample, you have to go in the lab, spread it on a plate, you cross your fingers, you hope something grows. And then you try to determine which species of bacteria or yeast that is. But you can miss a lot because some bacteria are just not culturable, so they don't grow in that environment, and they do adapt fairly quickly. So if you change their environment, then you are potentially missing a lot of interesting information about them. So with this new approach, you can really study them in their natural setting. And again, like right now, this is still in the lab as like a proof of concept, but you can imagine a world where we could use this to edit the microbiome or use it in soil, to edit soil microbes around plants for better, carbon storage or better uptake of specific nutrients. There's a lot of possibilities there.
Yeah, wow, that's interesting. Yeah, it's kind of like, yeah, once we have the new tool, then we can start updating the tool and be using it in all these scenarios. And as you know, it's like some of the, it makes me think too, actually. And the question I was going to ask 2 seconds ago was, you talked about for plants, you can take the, you kind of made it sound like this kind of brute force thing where it's like, okay, you can like chop them off and see if these plants do well and these don't do well. And I agree with that. But there's also that, what that just makes me think of is like, oh, if we're brute forcing something, can we just use AI to do the simulation brute force ourselves? Do you see much of the, like, are we doing, does AI, how much is IGI doing AI adjacent things, AlphaFold, blah, blah, blah?
Yeah, I think there's one researcher that is using AI as part of these CRISPR screenings, but I'm not entirely sure what he's doing. It's not my area, and I'm not as well versed
Yeah, you're good. And you can
in
see my
AI.
question was also not well versed either. I was like, blah 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 AI. So let's just pretend, yeah, we'll just come back to that later. The, so yeah, so we have CRISPR, and which is these little cut and paste scissors, but can also be or yeah, you can in addition to understanding what the genome is with sequencing and then understanding how to create new kind of DNA with synthesis, we now have CRISPR, which allows you to kind of find a little place in the genome and just kind of insert some new DNA there or cut some out. So that's obviously a big deal. And you're using it with public health and some of these initial kind of disease things, but also with climate and trying to help us with climate. And then we're obviously upgrading the tools themselves. And so for you, Melinda, you've been doing this at Gates, you're doing this before as well. How are you and how is IGI thinking about public impact here? And specifically, maybe I would want to ask, like, what do you see as the biggest opportunity with CRISPR, and what do you see as the biggest kind of challenge?
Mhm. Um, So I would say, one of the biggest opportunities, I mean, we have this amazing tool that allows us to change the DNA of, any living organism in a nuanced, targeted, and controlled way. And that is really powerful. So that's kind of wow. I think some of the biggest challenges um, is thinking through how to wield that power and doing so with restraint and with societal participation. Um, and I think when I think about this, I think about using the past to help contextualize what might happen in the future. I think the biggest issues with a lot of new technologies is developers are very much focused on the short term. So they're, can we do X or Y? Can we get this to work? Will someone want to use it? Right? That's usually where most developers are living, but they're not frequently thinking through, oh, what happens if everyone adopts this? And what happens if it becomes super wide scale? Um, then what would be kind of the ups and downsides to that? I think, I'm lucky enough to work with a lot of thoughtful scientists who are very much thinking about this already, but we all come to the table with a limited set of experiences and our own biases. So I think we really need to expand the range of people that we're talking to and really engaging with the public to be able to bring in more of that societal dialogue into our decision making. And that's always really difficult to do. It's like, who is society? How do you reach everyone? Who has the final say? So I think governments have a big role to play here as representatives of the people in, providing the guardrails. So to sum all that up, I would say the real opportunity is that we have this amazing, powerful technology. The real challenge is wielding this tool in an incremental fashion that shows restraint, and that's done with true societal buy-in.
Got it. And so that mostly makes sense to me, is that, yeah, let's use the tool and try to get because I'm reminded as I was doing research both for this show and just more generally with biotech, I was like, I learned about the Similar conference where we learned about recombinant DNA, and it was like, oh, let's like 1975, it's like, maybe chill for a second, like this could be really intense, and we don't want to like, change everything and make these crazy, scary designer babies or whatever. And then over time that we had some good restraint there maybe, but then there was also this thing of like, hey, let's actually use this and make lots of insulin for everybody or whatever. So and I think it's interesting you talk about like the societal buy-in or something like that. And that's kind of connected maybe to like the public education piece and the government piece. Is there, so like, maybe could you maybe like describe, that's still kind of maybe too abstract for me. It's like, what would you want to see in the world that would be like your optimal future, for that? Yeah.
My optimal future as far as societal engagement, or as far as what we're
Societal
doing with
engagement.
this technology?
Yeah.
I mean, in my dream world,
Yeah.
there would be this like amazing platform where scientists really do want to engage with the public and they really care, are able to like directly engage with a cross section of society. Like a Twitter for scientists and societal engagement would be ideal. And they can really hear feedback from people about like, here's some concerns, or here's how this might impact my community if this was to be deployed here, and be able to sort of aggregate all that information and use it to help us make better decisions about what to pursue and what not to pursue. I mean, right now we are guessing at what societal values are, or we're using our own values. I mean, that Asilomar conference was amazing, but it was also like a bunch of scientists sitting together and saying, like,
Yeah.
here's our values and here's how we should proceed. So how do we get broader representation into that decision-making process? Um, I think is something that would be incredibly valuable and useful, and that doesn't quite exist yet.
Yeah, yeah. It's interesting because if what I'm hearing you say is that and we almost want that same thing, but not just for biotech and CRISPR, but for everything. It's like, hey, there's going to be a decision made. How do we know? And that decision is going to impact some people. How do we get them? And I'm writing with some of my friends who work in urban planning, and it's very, I don't know, hip these days or whatever to do. I forget what they call it, but community engagement surveys or whatever. Where they're on the ground, they're trying to chat with people, they're trying to like drum up and try to get folks who are going to be impacted to kind of see what do they think, and so yeah, I could see that being so I think I'm mostly convinced and excited by something like that, which is and I say this because like, oh, what do I mean here? I guess there's like, yeah, no, I think that is that would be an amazing future. Especially, I think that a beautiful part about that is this like connection between scientists, like the ivory tower, and the people, where you just got these scientists who are just into their research, but they're happy to chat with the random folks and answer questions and do whatever, and then the people like get to actually have a connection with the scientists and like ask them like, what's going to happen with this new drought-resistant thing in my community or whatever. Okay, that makes some sense. Do you see? Maybe a question for you is like, how much of your work is also like, do you imagine this being put like, will there be regulations and laws, and what will that all look like around CRISPR technology?
Yeah, I mean, I think, regulations and policies are always lagging, right? So usually something happens and then the government's like, "Oh boy, we need to step in." And then they kind of step in. So it's usually like coming a little bit behind the technology, which is like totally normal. I think on the environment and agriculture side, we have heard sort of clear direction from the US Department of Agriculture. Last week, the IGI hosted a conference with the US, a whole bunch of different US government agencies. So, these new biotechnologies are regulated by the FDA, the USDA, and the EPA. So we hosted this workshop where we talked with all of them to say, okay, if I was to edit a microbe and put it into the environment, who should I talk to and what would you be concerned about? And so we had this like pretty amazing dialogue with the regulators, where they were walking through specific case studies of like how they would think about these things, which I thought was really good. On the human health side, there's all, the FDA also regulates and pays close attention to what's happening in the space and are asking questions about, yes, the safety, but they also want to see this technology advance because they
Mhm.
see the promise. And so they are actively engaged in setting up platforms to help companies and organizations like our own be able to get through that regulatory process and get things into IND. So um, it's really good to see at least the US government is really thinking about this and trying to move forward. Globally, the picture is quite disjointed. Like different countries are at different points in their regulatory process. So I think just last week, China said that they would allow some genome edited crops. So we're seeing things happening, but it is like a bit fragmented.
Yeah, that's interesting. Well, it's cool to hear because I think, especially in my more kind of crypto libertarian circles, everybody's like down on the like it or just the regulation with the certain things. They're like, oh man, it's tough. But the way that you talk about the regulatory bodies here seems awesome. They're there. They want to see it be positive. And they're also like, let's make sure it's good, and let's like be on the cusp of these new technologies. So that just sounds like overall like a very good relationship. And thank you for hosting that cool conference with them to
Yeah.
talk about it. Um,
Yeah, I mean, I think we see regulations as being incredibly important
yeah.
to um, the responsible use and deployment of these technologies. Like we see it as a part of the journey.
Oh yeah. Yeah,
Yeah.
definitely,
No,
definitely.
no.
Yeah, and I think and it's interesting because I think that the first CRISPR tomato, there was like a tomato, a GABA like tomato in Japan that's out there and it's being used.
Yeah.
Is there anything specific The interesting thing with regulation too that I've kind of found over time is that it's like, oh, here's this new technology. It's so crazy, and we're gonna have to change the rules or whatever. It's like, well, actually, the old rules just still kind of apply. Of like, you look at the harms, you look at the benefits or whatever, and you just apply it with the new technology. Is that true here, or is there anything with CRISPR that's specifically like, ooh, we gotta like make sure on this thing or whatever?
Um, I think it partially depends on what you're making.
Yeah.
I think for the most part, there are pretty extensive rules for genetically engineered plants that gene edited crops fit within the framework. I think a lot of regulators globally have been saying, um, these products are similar enough to something that could have arisen in nature on its own. Like if you're just making one base pair mutation, that happens all the time. And so they're saying, if it's so similar that we can't tell it apart, then we're probably not going to choose to regulate it. I think that does raise some concerns. You could make, a few base pair changes and make something that has an allergen, for instance. Um, but I guess in that case, yeah, the FDA would catch that in the US. So I think there's pretty good coverage across the board. I think for some people who want to avoid these products, and there is definitely like a segment of society who, they don't want genetic engineering or genome editing like near their food, and we need to be respectful of that segment of society. I think if you ask someone from that segment, they would say, we can't test for these. So, like the old GMOs, you would use an agrobacterium, a bacteria, to shuttle in the genes that you wanted in there. And so you could use that agrobacterium as a marker to be able to figure out what was genetically engineered and what wasn't. With genome editing, you're making these very targeted, sort of scarless changes. And so there's no way to really tell. And I think that, if companies don't have to disclose necessarily. And so I think that that would sort of violate the values and norms of that segment of society. Um, so that might be one thing that would be sort of different and fall outside the bucket.
No, that's interesting. I think all that is fascinating. That we've been doing GMO stuff for a while, and so a lot of this kind of fits in that bucket. And then, as you said, sometimes if the thing kind of looks like it, if you're just doing a little change, like this could have come from nature, could have come from like people doing this. It's like okay, this is probably fine. And as you said, sometimes a small change in the gene can actually have a big change in the phenotype or whatever. So who knows? But and then, as you said, also the scarless. Yeah, that's an interesting term for it's like, oh man, how do we still how do we kind of disclosure policies of those kind of things where we're changing these the genome itself without who knows how it got changed. It could have been us, it could have been nature, who knows. So I want to kind of, as we get into pseudo-ish, closer to wrap mode here, I want to ask you maybe kind of a wider scope question, which is more connected into my book and kind of the future of the human race or whatever. So I'm writing this book right now just to give you some context around like what information wants and the history of genes replicating and making the tree of life in the biosphere. And now we have memes which have replicated that have made religions and companies and money and all these, like the idea sphere, and also all the tech in society. And now we have humans are able to actually edit the genes, these replicators. And so how do you see a thing that I'm trying to understand? I don't quite grok yet. It's like, how do you think humans, as we start to use CRISPR on ourselves and on nature with the roots and all these things, how do you see this happening in the next 25 years or whatever? Us changing the biosphere, and changing genes.
Yeah, I mean, I think ultimately, and I don't know if it's in 100 years or 300 years,
Yeah, yeah.
I don't think we're going to be limited by the science. It's going to be more around our ethical, moral, and societal constraints. Um, and then, as far as what I think is going to happen in the next 25 years, going back to my comment around history, as humans, we've evolved quite a bit. We've made massive changes to our environment. We have lived through multiple technological revolutions, and I would say we are living in one right now. And I think of the technologies that have really permeated and integrated themselves into our society, like the printing press, light bulbs, the internet. These things fill a real need, and they make our lives easier in some way. I think after they have reached widespread adoption, they start to raise new issues based on just the fact that so many people are using them. So, my prediction is the first things are going to be in places where there is a real need. So, is there like a genetic disease that's causing pain and suffering? Is it cancer? With climate change, we can see some of the crops that we heavily rely on for food that have been bred to have fairly little diversity. So things like banana, for instance, and the bananas are threatened right now by a really bad disease. I could see those being kind of the low-hanging fruit, not to make a
Nice.
pun, of things where, like, in the near future, the only banana you might be able to get is an edited banana, right? And then further down the road, I think it's going to be things that there is still some kind of, like in 100 years, I think it will still be things that maybe are somewhat preventative, like thinking like Alzheimer's or other neurodegenerative diseases. But I think it's mostly going to be practical for the short to medium term.
Yeah,
Yeah.
as you said, it's things that complete meet human needs are the things that we want and that that society pays for. So it's like, yeah, if the light is good behind me because it is, and I pay for it because it meets my need for light. And similarly, as you're saying, yeah, all these like disease things or food things, it's like if we have those needs, well, then the kind of money will go there. And it's interesting. I like what you're phrasing around the constraints for the long-term future, which is why it's so important to both democratize access and those kind of things. It's like what do we value, and that's kind of what will happen. It's not really the science that will constrain us, it is us that will constrain us. So cool. Well, that's interesting. And I think maybe these final two sections then are I guess do you have any advice? I think you've done a good job of both leading a science career but also leading a kind of a public policy career. What advice do you have for young folks to lead an interdisciplinary career like yours?
I would say just try things out. So I think I definitely approach my life and my career with like a sense of curiosity and trying out lots of different things and then trying to smush the things that you like together. I think that is usually a pretty good approach. I would say that I've had to get very comfortable with being uncomfortable. Normally I only understand like that much. I'm like not of any one thing because I'm doing like 10 different things at once. So, you have to be okay not being the expert in the room and approaching your work with a sense of humility. And in that sense, it can be really fun and rewarding.
Yeah, no, I love that. I think being comfortable with being uncomfortable. It's like, yeah, you're going to be dumb in a lot of different ways. So it's like, what's happening over here? What is this to do with the AI and the attention? Whatever. So, yeah.
Well, I just have to keep reminding myself that there aren't a lot of people that do work across all these different areas. So in some sense, you are special in your own unique way,
Yeah.
but you're definitely not an expert in any one particular thing. And that also comes down to temperament and values. Some people do derive a lot of personal satisfaction from being like the person that knows a lot about this thing. And so it's also a matter of like what really fits with your personality.
Yeah, no, I like that totally. And yeah, and for us, the kind of wideness fits with our personality versus the depth. But depth people exist too, and some of my best friends are depth people. I have.
Me too.
One final question here is usually at the end I do like a little overrated, underrated thing. But for you, actually, I want to ask one kind of variant on overrated, underrated, which is, and you might be biased to answer this a certain way, but if you were to rank the importance of like sequencing genomes, like genetic sequencing versus like synthesis, the like active creation of DNA versus like CRISPR style kind of cut and paste or whatever, which one, what
Mhm.
would be the like importance of those three? How would you rank order them?
Mhm. I would put sequencing first, mostly blasphemy.
Yeah.
Mostly because, sequencing is what helps you understand what's actually there, right? Like you need to be able to read the book to know what's in it. And CRISPR wouldn't have happened without sequencing. They sort of build on each other. And then I would say genome editing would be the second thing. I think it's much easier to go in and like make small changes to something that already exists than to do something like synthesis where you're de novo trying to make something new. So I think that's how I would rank them.
Beautiful. That sounds good. You and I thought you were gonna rank editing top, but no, sequencing top. So that's good, or at least a very a political, not a political play, but a very you're making sure you love all the people, the different kinds. Beautiful. Well, thank you so much, Melinda, for your time today. Is there a place that folks who are interested in kind of CRISPR for good or whatever, is there a place that they can either find you or find kind of IGA's work on the internet, or any
Yeah,
recommendations for them to go forth in the world?
of course, the Innovative Genomics Institute.org, we have a ton of excellent educational resources. So if you click on the drop down educational tab, lots of accessible videos for people at all levels to understand more about genome editing. It's a really good place to go.
Beautiful. Yeah, I mean, I think it's a great, and I'll put a link in the show notes there, and also to Melinda's bio. And I do think that there's a cool, yeah, if you're looking to either get into kind of biotechnology or CRISPR work, this is a cool way to both have, if you're looking to do research, but also research that has like, kind of like publicly applied research. I think it's a cool spot. So with that, Melinda, thank you again for your time
Thank you
today,
so
and
much.
thank you,
That was
listeners.
fun.
Goodbye, everyone.
Bye.
Thanks so much for listening today. If you liked the show, please give us a 5-star podcast review or subscribe on YouTube. And if you'd like to chat about this episode with a community of amazing, smart, ambitious, divergent people, come on by and join our Discord. You can find it at root.co. That's ROOTE.co. And then finally, if you'd like to contribute to these ideas being shared more widely in society, you can support the podcast production team at patreon.com/Rhys Lindmark. That's patreon.com/RHYSLINDMARK. Thanks, and see you here for the next episode. Bye.